Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins DiscoverX, I'd like to thank you all for attending. I'm your moderator, Ellen Capon, Editorial Assistant at Drug Target Review. Today's speaker will be Terry Kenakin, Professor of Pharmacology at the University of North Carolina School of Medicine. Following their presentation, we will move on to a live question and answer session. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to Terry.
Well, hi folks. Welcome to the webinar, Value of GPCR Cell-Based Assays in New Drug Discovery. My name is Terry Kenakin. I'm a professor of pharmacology, UNC School of Medicine, but I've spent most of my career in industry. So I will present this talk from the point of view of chemists, biologists, moving compounds forward towards drug candidate selection. And in that process, of course, cell-based assays have been and will be important. Mainly, we know them for finding off-target effects, safety issues. I'll mention these, but today I'd like to expand on them and go beyond them and tell you how cell-based assays can be used to define mode of action and hopefully to characterize therapeutic value. But just to return to the standard points of target effects, we all know that we want to know what our compound does on enzymes, ion channels, other receptors, and so forth, and assays are used to do this. These assays have to be comprehensive, accurate, and sensitive. Historically, binding assays have been used for this. They're simple, low cost, but they furnish incomplete data, and you can miss a great deal simply using binding. Technology has now afforded us using cell-based assays, and these are the main ones that are important now in that process. They can find agonism, allostery, biased signaling, and a whole host of other complex effects on the cell. In terms of safety issues, we will use animals, of course, to assess possible toxic effects of our compound. And the first question to ask before that will be, what is the compound effect on the animal receptor? And so here is again where cell-based assays are used. They can be used to address this question. Historically, orthosteric compounds that interact with the natural hormone or transmitter binding site don't show very much variation across species, usually because the chemicals that bind the site are the same in animals and in humans. So you see fairly good correspondence. That is not the case with allosteric compounds. They can bind anywhere on the receptor. This just shows a few sites for the known allosteric molecules, and here you can get great variation between animal and human receptors, and so it's critical to test the animal receptor with your compound. An example here is an allosteric compound for mGlu1. You can see it's active on the rat receptor, but not active in human. This can have complicating effects on development. Here we have a compound. It's a positive allosteric potentiator. Dopamine is potentiated. The curve shifts to the left. And this type of activity could be useful for increasing cognition in schizophrenia. And this is data for this compound on the human receptor. However, when you test it on the rat, there's no effect at all. And what this does is really preclude further testing, because this kind of compound should be tested in a rat novel object recognition assay, but of course that is not the case here. So very important to check animal receptor activity. But now let's go beyond that and start with asking questions. How can cell-based assays be used to determine drug mode of action? Again, easily we can test compounds and assays on cells, determine if it's an agonist, an antagonist, and an allosteric compound. But we now have a greater array of assays that can really narrow this down and give us texture, what kind of effects we see. And these are the effects that will give us the ideas about what our compound will do therapeutically. Of course, what we see depends on what we look for. And so when we're looking for efficacy, very important to note what assay you will use. Michel Bouvier coined the term pluridimensional. What he means here is that drugs have many efficacies. And so as you test compounds in various assays, like an Escher print, you will uncover different activities. And this takes efficacy from what it was historically, which was thought to be a single signal varying only in strength. We now know that efficacy has quality. A little bit of GS, GI, beta-arrestin, and so different drugs give different qualities of efficacy to cells. And this is where the therapeutic richness comes in to the program. And we all know that drugs have many efficacies. What can this do? Well, theoretically, beta receptor inhibition should be a good thing for treating heart failure. And because of that, 16 beta blockers have been tested in trials for heart failure. Of those 16, 3 worked well and 1 worked best of all, carvedilol. If it was that single property, why didn't they all work? And the question may be that the efficacies of carvedilol, shown here, are the important aspect for the overall therapeutic outcome. And so maybe it's the whole collection of these that we need to assess to get therapeutic value. And of course, the main tool to do that will be cell-based assays. They are the key to finding all efficacies of a compound. All this coming from GPCRs, they are the creators of all these efficacies. I call them the Swiss Army knife for physiology. They have a lot of functions and they can affect cell function in a great many ways. We all know they interact with many G proteins, so we have that texture. But now we also know that they can interact with beta-arrestin, which can form scaffolds for many kinases and so forth, producing a further array of signaling. They can also put the receptor in various parts of the cell cytosol to give unique signaling as well, so that they have a great repertoire of producing changes in cell function, and all of these, or many of these, could be relevant for therapy. Just now to change gears and talk about molecules binding to a receptor. Dynamics tells us that affinity and efficacy are correlated. The same forces that control binding of a compound to a protein also control the changes in the protein structure when a compound binds. And this happens with all molecules. So don't think of binding as being a passive process. You can bind a molecule to a protein and it'll stay the same as it was. No matter how large a protein is and how small the molecule is, the protein will perceive it, and so its energy will change. This really has changed our view about efficacy. Historically, this was what was thought, that of course all drugs have affinity, and then some are special. They're agonists, and they have efficacy. They produce cell function. This was thought to be special only because we had limited assays to see what happens when a drug bound. Now we have many more types of assay, and we see there's a lot of efficacies going on that we would not have seen with a simple assay like cyclic AMP and calcium. In view of the idea that proteins change when you bind a compound to them, you could rephrase this and suggest that almost all drugs, when they bind, will have an efficacy as defined by the change in behavior of the receptor towards the cell. So if you run an assay and you don't see an efficacy, it just could be that you're using the wrong assay. Running a screen, of course, you get your hits, and let's call this hits, call these hits a pot of gold. I call them pot of gold because these are molecules that bind to the protein and as such may indeed change it. So if we again define efficacy as what changes the target behavior towards its host, which is the cell, and many of these compounds could have efficacies, and the only way to find that out is to test them in various assays where you can see it. I now like to talk about two aspects of efficacy in terms of detecting it and how it can fool you, and that is they can hide behind differences in cell sensitivity and biased signaling, beginning with sensitivity. So agonism, that is efficacy you can see, is a partnership between efficacy of the compound and tissue sensitivity. So if you have a compound of low efficacy and you test it in a tissue that is not sensitive, you may not see anything, which is fine, except if that same compound goes in vivo and it meets a highly sensitive organ, you may get a surprise and see an agonist effect that you weren't counting on. So again, if you're trying to characterize a compound, it may be important to know this. Think of efficacy as a battery here. There are compounds with high efficacy, and those are the ones with large batteries and smaller efficacies. And these drive machines. These are the organs in vivo. And a high efficacy compound will probably produce response in all tissues, but a low efficacy compound may not. So this is what you see with a compound with high efficacy, like isoproterenol. You see strong response in all tissues. Tissues are shown here. Whereas a compound of lower efficacy will not produce response in all tissues depending on their sensitivity. So it's useful to have a type of volume control, and the usual one is level of receptor expression in a cell. So you can have tissues of low sensitivity or high sensitivity, where you can hear Led Zeppelin here. Here's what can happen. This is the testing of carbachol and oxytremerine in a tissue of low sensitivity. And what you see is you see the curve to carbachol, but you don't see a curve to oxytremerine. Same drug, same receptor, more sensitive tissue, and you see that oxytremerine has a very powerful response in this issue. So this is the kind of thing you want to avoid. If you don't want efficacy, you need to test compounds in this kind of assay. Again, cell-based assays are the key. And that's just restated here. And finally, biased signaling. We know that receptors activate multiple signaling in cells. We now know also that they don't do it in a uniform manner. This is new. It's called biased signaling. It's only new because we just have the means now to measure this. It was always going on because it's a standard outcome of allosteric protein function. So what do I mean by biased signaling? If we have a cell and we activate the receptors with angiotensin, in this case, we see GQ protein response and beta arrestin response. We can take an analog of angiotensin, in this case Trevena 120, same cell, same receptor, and you will only get one of those. So Trevena 120 will only produce one response. It biases the signal. How does this occur? Receptors can form numerous active states, and different ligands can make stable different active states. And so we can have agonist 1 could make a state that might interact with G protein 1, whereas agonist 2 may make a state that will interact with G protein 2 and beta arrestin and so forth. This is a very useful therapeutic tool now, and it's being used to fine-tune signaling. It can also be important when you're looking for efficacies of your compound in cells. As I said, it's an outcome of natural allosteric probe dependence. So if you look at this panel here, panel A on left, we see a protein and it interacts with two tracers, two things that interact with it. We add an allosteric compound, changing the shape of that protein. There's no rule that says the effects on tracer 1 and tracer 2 will be equal. In fact, the experience is it rarely, if ever, is. This is what is called probe dependence. All proteins do it, enzymes, ion channels, and especially GPCRs. Well, if I take those sites, tracer 1 and 2, and I put them inside the cell and give them names, call them G protein site, beta arrestin site, that is all bias is. Standard allosteric probe dependence, and it happens for all receptors and all ligands. So consider it like the tip of the iceberg being the allosteric nature of GPCRs. And so, whether your program seeks bias or not, synthetic ligands most likely are going to have a bias different from the natural hormone or the transmitter. We should not expect that our new compounds should be copies of natural agonists. I'm often asked whether a program should seek bias or not, and the answer is no need. It will seek your program. It's there. It'll happen, and so you should not ignore it, because most new agonists are biased. Here's an example. This is tirzepatide, GLP-1 agonist, diabetes, weight loss, and it's biased, just like most new compounds are. So this is an important aspect of your compound profile, and the best way to detect it is with cell-based assays. A frequent approach to this is cross-screening. And so when a screen is run and I'm showing activity as these points to the right, these are the active hits. The history of this has been you take your hits and you test them in your next assay. Now, most often a wider swath of hits are cross-screened in a second assay. And so these are responses of these compounds in assay 1 versus assay 2. When you do this, I guarantee you will see a scattergram. You will not have equal activity. Now what I can do is move the compounds as exemplars shown here into the next assay. Why would I choose to do that? Because I know they are making stable different active states. They are different. Whereas these guys, I don't know. They may be all doing the same thing, so I won't see any texture here. These compounds will give me the highest chance of getting a unique phenotype, and so you would advance these compounds from across screen. So we're going to end this with talking about a few case studies. And it's a question of trying to take the snapshot, which is your in vitro assay, and predict the whole movie, which is your compound going in vivo and activating a host of organs. And the link between these is the model. I'm going to show you an example of where this was done and where it was successful. This is an allosteric potentiating compound, BQCA. It potentiates acetylcholine, and so the acetylcholine curve shifts to the left. But when tested in 4 different cell lines of varying sensitivity, we see different profiles. Here we see the shift with agonism. Here we see shifts only, and here we see an increased maximal response. This came from a real case study, the chemists were, of course, confused. What should they use for their scale of activity, changes in baseline, location of the curve, and so forth? And so they had literally 3 different scales of efficacy. What you can do in some cases is apply a model to try to get at the underlying behaviors causing this. And so we link what we see through models. Some are better than others. This is the model of efficacy when Kenakin was a grad student. But as Hawking points out, the best models have rules. They are you can fit equations and predict behavior. Once you have an equation for a system, you can predict the whole universe of what that system can do. So this is the aim, and in this case, we were able to fit the BQCA curves with the functional allosteric model shown here, which is the Stockton-Ehlert binding model as the front end of the operational model. And this equation will predict what you see and give you parameters of what might be happening at the level of the protein. And so you get parameters alpha, which is changes in affinity, beta, efficacy, and so forth. And when you do this, you can take these different cell lines and fit to these models to give you these. These are supposedly what's happening at the protein. We were able to get a nice uniform set of parameters, and this takes away the variation of the cell lines. So now the chemists did not have to worry about different profiles they saw. They can simply see that the compound, in this case, BQCA, increases of affinity 15-fold, efficacy 2-fold, and has a 1.4% direct efficacy. So cell-based assays are the key to this kind of analysis. Finish this with some case studies. Nothing worse than spending years on a compound trying to get everything right, putting it in humans, and it fails. And yet we have between a 50 to 80% rate of attrition because of efficacy for new drugs, which is really quite high. Think about that. This is where teams did everything they thought they knew how to do, and the compound still failed. And you could discuss this in terms of not knowing enough about your molecule before you went ahead and put it in humans. Here we have a failure, and perhaps he's just saying, I should have known my molecule better. Why would you want to do that? Well, again, molecules are much more complex now. We know this, giving these different qualities of efficacies to cells. And we're probing very complex physiology. So with these, the more we know about our probe, we can gain an insight into the disease state that we're trying to fix. But also, if our molecule fails, we don't want to follow up with a compound that does the same thing. So the more you know about your compound, the better. You can read more about this. The examples I'll show are from an article. This is an extensive article describing where molecules were put in late stage and failed because of nuances in efficacy and various properties. So, and that can be found in that paper. So let's talk about a few examples, and there they are. Some are real, some are hypothetical. The first is real. H2 inhibitors for the treatment of ulcer failed upon repeat chronic dosing. And so let's make an example of this. We have a compound, loser 5974, and it works acutely, gives a good positive ulcer score, but upon chronic treatment, the effect goes away. What this team should have done before they went ahead with this compound is determine if this was an inverse agonist. So why would you do that? Because an inverse agonist can cause increased receptor expression on a cell and negate inhibition. And this is what happened with the H2 inhibitors. Tolerance was observed upon chronic treatment, and it was shown that cells greatly increased the cell expression of H2 receptors, because an inverse agonist prevents the spontaneous activation of the receptor. And cells have a wheel. They produce receptor, it's expressed on the cell surface, it becomes phosphorylated and internalized. And so different receptors, different cells have various speeds of the wheel, and you have a steady state until an inverse agonist stops it. And then the receptor, the cell is still producing receptors, and you will get tolerance. So it's a good idea to know if your antagonist is an inverse agonist for that reason. Failure to follow up possible allosteric probe dependence. I'll show you an example with GLP-1 published by Cassandra KOL. GLP-1 has 4 natural agonists. Some are more important than others. The most important one is shown here, GLP-1 736. And their GLP-1 PAM Novo 2 produced very little effect here. This is probe dependence. A minor agonist of centromodulin was showed a huge effect, but therapeutically this wasn't important, and so they should have tested all probes before going forward. Failure to detect changes in cytosolic translation of receptors. A good way to prevent HIV infection is to get rid of the receptor that binds HIV, which in this case is CCR5. So chemokines can be used to internalize this, thereby not giving HIV anywhere to bind. And two chemokines that do this are ALP rantase and rantase. However, you can see the receptors internalize, but if you go further and do other studies, you need to follow what happens to the internalized receptor. And in this case, rantase internalizes the receptor but lets it rapidly emerge again. And so what you get is the receptor goes in, comes out again, and has no effect on HIV binding, whereas ALP rantase will internalize it and the receptor is destroyed. Failure to differentiate allosteric effects on affinity and efficacy. So here's a case where you might want to use a potentiator, an allosteric compound that potentiates response for a failing system. And so something like Alzheimer's, where you get cholinergic transmission begins to fail, you might want to use an allosteric potentiator of acetylcholine, and we have one here, add 7973. It potentiates the curve to acetylcholine, just like we saw with BQCA, and so should be good for increasing cognition in Alzheimer's. Cholinesterase gives a positive cognition score. Bad 7973 did not. What was wrong with that? It should have worked. Well, had the team looked at the effects on affinity versus efficacy, they might have shown that, understood that bad really would not work. You can increase response two ways: increase the affinity of the receptor, the efficacy. In a failing system, changing the affinity really does nothing. And so in this case, this compound increased affinity. It did nothing to efficacy. This is what you would need for an Alzheimer's drug to increase efficacy. And so as we showed with BQCA, we had tissues with varying volume control, high sensitivity ones and low sensitivity ones, and should test the compound in this assay to see the effect on efficacy. And finally, failure to detect low-level agonism in PAM. This came to the fore with GLP-1 allosteric compounds for diabetes. GLP-1 agonists like exenatide are used to potentiate release of insulin through GLP-1. What happens when someone eats a meal, GLP-1 is released, and that will increase the insulin release. An agonist beats away at the receptor constantly, and what was happening with these is nausea. So the idea is to use an allosteric compound that does nothing until a person eats a meal. And so when the system is activated, then you got the increased response. You had an intermittent effect that was therapeutic, but it wasn't constant, and the hope was you would not get nausea. So here we have a compound, and we'll call it week 8915, and it potentiates GLP-1. So if we test it in people and there's a nausea score, we see that agonists produce nausea, but so did week 8915. So what was the issue here? Perhaps this compound should have been tested in a sensitive cell line where direct efficacy could be determined, and that's shown here, and that might have uncovered the property. So that's the end of the lecture. You could get drugs from a screen, run your curves, get their potency, and stop there, but you would have no texture as to what these molecules do. If you apply cell-based assays and study them further, you might get a lot more texture, and then you're better equipped to see, to relate what you see in clinical trials with profiles of compound. And the key is having the assays. And so if you have GPCR resources, large collections of cells, receptors, or ways you can change cell sensitivity, this goes a long way into determining the quality of efficacy of your compounds and detecting activities you may not want in a compound. So with that, I'll stop and see if anyone has any questions.
Thank you, Terry, for your excellent presentation. We now have the pleasure to introduce our questions and answer session. Don't forget you can still submit your questions using the questions panel in the menu on the right. So let's take a look at the first question. I'm performing CRE report assays for several GPCRs. I want to obtain information about selectivity. However, the Lodge IC50, a compound in the CRE assay, is very much dependent on the ligand concentration. How can I do a fair comparison?
Well, hi, folks. Very nice to meet you all, virtually anyway. So I assume an IC50 means an antagonist, and you're seeing variation with concentrations of agonist. Doesn't matter, same idea with an EC50. The whole key in this is comparison. So you always compare to a reference. There are correction factors if you know how the drug works. If you have a competitive antagonist, then you'll do the Chang-Prasov correction for the agonist concentration, and you will get the affinity. Sometimes, though, you may not have this, so it's always a good idea to just have a reference compound, compare everything as ratios to that. One other quick comment on that: a competitive antagonist will vary more with conditions than a non-competitive. That stays pretty rock solid, and you shouldn't see that kind of variation. And then if you're using allosteric compounds, the same thing holds. If you have an alpha NAM which affects affinity, you'll see more variation than a beta NAM which affects efficacy. I might add, if anyone asks a question and I haven't really covered it well enough, email me and we could take it offline.
Great, thank you. Our second question, can a PAM behave like an NAM at times? If so, what is the explanation?
All right, let me get that one. Can a PAM behave like a NAM? PAM and NAM are general descriptors. PAM generally means you potentiate response, and there's various ways to do that. Affinity and efficacy, as you heard. NAMs do the opposite. But there's no rule. An allosteric compound can reduce one and increase the other. And a very interesting class of compound is the PAM antagonist, which increases the affinity for the agonist, but decreases the efficacy. These guys become very special. They actually get more active the more agonist is present in the system. So they give you an extremely unique profile. But I'm assuming you mean with different cell types or conditions or sensitivity, can a PAM suddenly become a NAM? And it shouldn't. If it's doing something unique to the affinity and efficacy, that will translate in pretty well any system. I would point out, if you have examples or data that shows this, I would be very interested to see it. You can email me on that too, please, for my sake.
How would you quantify bias, taking into account both the efficacy and potency?
Great question, because most early on, people simply used potency, IC50, but agonists behave differently with changes in sensitivity when they're partial versus full agonists. So you have to somehow account for the maximal response. The operational model defines a way to do this, and so and this has been published quite often now. Log tau over Ka, which is efficacy divided by affinity, and that you can approximate that with maximal response divided by the EC50. Notice, if you're comparing two full agonists, that's just a ratio of EC50s. So this new scale only kicks in when one of the compounds does not produce maximal response, but it does correct for it. And there are articles out there describing it. There is an excellent review on the various ways people quantify bias by Ongun Onaran and Tommaso Costa. And then I've written some things too. So, but it's generally you would use logarithm of maximal response divided by the EC50.
Is it possible to detect cooperativity in the binding assay? We are running MGI-URs for binding and gamma GTP and CAMP functional in membranes and CAMP in cells.
Oh, yes, of course. The history of allostery, when you go back to the 70s, Monod, Changzhou, Wyman, they used binding. They saw cooperativity, and that's in fact how they detected and defined allostery. I'm not a big fan of binding. You can miss a lot, but you can also gain things that you can't see in function. But function is really what's important. And in terms of the trouble is, binding is probably a more direct cooperativity effect, or will detect cooperativity at the level of the binding to the protein. And so things like dimers and so forth, you'll detect them there. Function will give you differences in slope. So if you run a curve and your hill slope is greater than 1, less than 1, that could be cooperativity, but it's also the cell can do this for you. So it is not a clear look at what's happening at the protein. But I don't know of many allosteric systems studied with binding that do detect binding cooperativity unless you start to invoke some other species coming in, dimerization or some other things like a ramp binding to the receptor and so forth. But it should be possible. The whole key, as Hawking said, the equations predict the whole universe, and most of these models, they don't account for it. So if you see it, it doesn't fit with the model. It's screaming at you.
How would one know if we're accurately modelling a high versus low sensitivity cell model in vitro compared to in vivo systems, especially when the target of interest may have a different expression profile depending on the tissue?
Okay, Ellen, I'm going to have to read that one because that was a lot. Let me see, which question was that? Oh, okay. How would one know if we're accurately modeling a high vs low sensitivity cell model in vitro compared to the in vivo system, especially when the target of interest may have different expression? That's the whole point. Great question. Yes, yes. That's this idea of doing an in vitro assay. You're sort of chained to the system you have. So you have cells at a constant expression level and so forth. And the key is then you give your compound in vivo, it'll meet all kinds of organs of various sensitivity. And so you need to have a way to predict what could happen. The most facile way, and I'm seeing this more and more in companies and so forth, is to have a high and low expression system, just 2 cell lines. Run your compounds in each, and between the two, if you're fitting the operational model, you will be able to quantify relative efficacy. And then, like Hawking says, you can predict the whole universe. You can predict what your compound will do in reference to a reference compound in any system. So I tend to prefer a low sensitivity system because there you get a direct measure of affinity with the EC50 if you have a partial agonist, and you get relative information on efficacy from the maximal response. So your question was, how would I know if we're accurately modeling a high vs low? Oh, you're in space. You don't know in terms of what'll happen in vivo, but you try to get 2 snapshots of extremes, and then if you can fit equations to those, you should be able to predict any system.
For lead optimization, is it necessary to have more than one SAR assay? Is it better to use Emacs/EC50 or the operational model for tau and Ka?
Okay, let me just get back to you. I apologize. I actually prefer to read the question so I can Okay, I'm sorry, here we are. For lead optimization, is it necessary to have more than one SAR assay? Well, you know, I'm a proponent of as many assays as you can afford, simply because of this idea that binding is not passive, so you may uncover. So in terms of, the answer is yes, if you want to look at your quality of efficacy. When I say quality, that is, you don't just choose calcium, cyclic AMP, just one system. You want to test what else your compound will do. And then the bias of that mix will tell you the efficacy of the quality of efficacy of your compound, because all drugs are probably giving cells a little bit different qualities of signal, and that's the key. In there could be the secret of good versus failed therapy. So I mentioned using low and high sensitivity assays, and that can be for the same signaling system, but also I would encourage, at the very least, looking at G protein signaling versus beta-arrestin. There are now ways you could look at internalization without imaging. You can use a biosensor and get that. So there's all kinds of new assays coming out. And if you have access to them and can afford it, it would be probably worthwhile to look at your compound in various ways like that.
How does the overexpression of a GPCR influence its response to modulator compared to endogenous levels?
Well, that's a good question. The level of expression has gone full circle. When I started in industry eons ago, the key was, oh, let's build a highly overexpressed, powerful signal, stable cell assay. And you lose a lot. When everything's a full agonist, all you've got to look at is potency, which is a complex amalgam of affinity and efficacy. You don't know much. You can get a whole lot more in a low sensitivity assay where you can directly look at affinity and efficacy. But again, these are useful snapshots. I wouldn't presume that you're going to build the ideal in vitro system that will reflect what you'll see in vivo. I don't. You might, but there's a good chance you won't. So overexpression will simply reduce nuances in signaling and probably be a lot less sensitive in terms of seeing the quality of efficacy than a system that is not overexpressed.
In PET, is this important to the binding?
So the question I'm reading here, in PET, is important the binding? Is binding important? Is that the question? I will assume we're talking about, yeah, well, you know, if you're doing PET, you know a lot more than I do about PET, but my understanding is that there's two ways. You have a radio labeled compound and you look at where it goes and how much of it goes there, or you pre-label your site and then you use your drug to reduce binding there. In view of the really interesting work that's coming out with diffusion restricted systems, drugs go into these systems and they're trapped, and so instead of having a normal offset with clearance, they will rebind and so forth. I'm not so sure if you see a PET signal in an organ, that's direct evidence that your molecules binding to the target. Could be, but it could also be trapped in a receptor compartment around it. But overall, I would say, yeah, it's important. If you have a high affinity, then the assumption is you'll see the binding in PET and be able to detect it.
I am performing CRE assays. Just the overexpression of the GPCR results in an inhibition of the CAMP signal. Is this caused by production of ligands by the cells?
That's a very unique explanation. I have not really heard that one in terms of the cell actually producing the ligand. So the overexpression of the GPCR results in inhibition of the cyclic AMP signal. That's an interesting effect. You know, well, I'd love to be able to discuss this with you, because if it's a GS receptor, then next question is, does it interact with GI, and does overexpression? The thing with overexpression, when we were in industry, it was often used, and then people would be critical, of course, and say, yeah, well, you've overexpressed to the point where your compound and receptor will turn on signaling that it would never do in vivo because you've so overexpressed the receptor. And that's true. That's this question of can you recreate physiology? And I would suggest no, you can't. But overexpressed systems are very good for screening, because now you can detect. Screening is just something else. The screening signal isn't, you see an effect and you assume that's what'll happen therapeutically. You see an effect, and all that tells you is the compound binds or interacts with the target. So if it is overexpressed, it may bind and cause a signal that you wouldn't ever see in vivo. I'm actually just rambling around. I cannot immediately see a way in which overexpression can reduce your cyclic AMP signal, but it interests me a great deal. Please feel free, Maria, to email me and take it offline, because I'd be interested in hearing more about that.
Unfortunately, that is all we have time for today. Please join me in thanking Terry for today's webinar. As you leave today, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins DiscoverX, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.
Bye, folks. Thanks.